I'm going to be talking. I selected a number of uh, collection of data to give you an example of how specific metabolites, such as phosphonucleotides, play a very fundamental role in the regulation of gene expression in trypanosomes, especially of VSG genes, which are important for the parasite immune evasion. This parasite relied heavily on the regulation of VSG expression for evasion of the host antibody response, and quite a lot of our work has been now trying to understand the signaling process underlying the control of antigenic variation. We stumbled on this a few years ago, about 10 years ago now, and we are really starting to get understanding how this process is controlled. Before there, let me just mention briefly here what my lab is doing. My lab is working on has two main focus. One is the fundamental biology. We're trying to understand signaling, nuclear signaling, organization of the chromatin in space, and how the phosphonucleotide signaling controls compartmentalization of telomeres and gene regulation in these parasites. The second is towards Chagas disease in vaccines. I won't talk about this. I won't have time to cover that. But you have been used quite a lot of yeast surface display, genome-wide yeast surface displays to identify vaccine candidates and drug targets and inhibitors. Trypanosomes, I guess you may have heard of them if you don't work in the field. They are caused by single-cell protozoan parasites, Trypanosoma brucei, cruzi, the parasites associated with the biggest burden on human disease in Leishmania. Chagas affecting primarily South and Central America, but has spread to North America, United States, Canada, and all the other places in the world, where the sleeping sickness caused by Trypanosoma brucei affects primarily African countries. So those are single-cell parasites. They are transmitted by the bite of a tsetse fly, and that can cause disease in humans. These parasites will cross the blood-brain barrier, causing severe neurological damage. They also infect cattle and with economic burden on the population. So these parasites have evolved a very sophisticated mechanism to evade the host immune response. Trypanosoma brucei do not infect cells. They circulate in the bloodstream, eventually goes to the brain, skin, fat tissue, but essentially they are outside of cells. So they express a very homogeneous surface coat of VSGs, variant surface glycoproteins, which are shown here. Okay, so those are proteins attached to the surface by GPI anchor. And what's important about them is that the parasite periodically switch the surface coat to evade the host antibody recognition. So every time during the course of an infection that the host assemble an antibody response against the growing parasite population, we will start to clear, the host will start to clear that population. Parasites that switch the gene expressing the surface coat are able to escape, and that process goes on and on and on. And this is possible because this parasite has a very large repertoire of VSG genes. And I'll get into that in a second. This is just to illustrate to you the surface coat is very thick, and antibodies tends to bind to the surface, and they are endocytosed and destroyed by the parasites. This is possible because they have this very large number of VSG genes. There are about 2,500 of them. Those genes are mostly subtelomeric. Here you can have in this left side of the screen the 11 chromosomes of the parasites. The VSG genes are indicated here in red. This is the chromosome. These are subtelomeric regions. It shows like a fork because the two copies of their chromosomes are different at the subtelomeric regions containing those genes. So it's a huge repertoire of VSG genes. However, they are only transcribed from specific sites in the chromosome, which are called telomeric expression sites. On the expression sites, the VSG gene is the last gene near the telomere. So you have a POL1 promoter, they are transcribed by RNA polymerase 1 rather than 2. You have expression site associated genes, 7 base pair repeats, VSG genes, and telomeric repeats. So I'm going over this because this will be very important throughout the course of this presentation. We also have metacyclic expression sites, also less gene close to the telomeres. There are about 20 of those sites from which the parasite can express a VSG gene, but only one is transcribed at a time. And the way that they switch the surface coat is by either transcriptional mechanisms in which they turn off one of the expression site and turn on a different one of the 20, or they can recombine VSG genes from the subtelomeric regions of many chromosomes into those expression sites that can also result in the creation of new genes. So it's a huge repertoire to evade the host antibody response. What's important for us is that these expression VSG genes are transcribed from an expression site body, which is outside of the nucleolus. Outside of the nucleolus, because they are, I mentioned this because they are transcribed by POL1, and POL1 are transcribing the forgive me, the nucleolus. So here you can see the nucleolus standing by in red for POL1, in green for telomeres. This is a combination of immunofluorescence and fish. You can see the active expression site being transcribed here, POL1 and the telomeric region. Silent VSG genes, which are all clustered together. We didn't understand how, but now we're starting to get a hand on what could be holding all those telomeric together to repress all the silent VSG genes grouped together in this compartment here. We know the active expression site, the POL1, recognize the promoter and transcribe through the VSG gene all the way through the expression site into the VSG gene. It's a 46 kb long expression site, whereas the silent ones are repressed. And some proteins have been identified as important in the regulation of the process, okay, of this process. For example, for the repression, which I'll be focusing on here today, we have we know that RAP1 plays an important role. NUP1 is the nuclear lamina protein. RAP1 is repressor activator protein 1, which is a protein conserved in yeast, humans, and many organisms with a role in the telomere silencing. And we identify the phosphatidylnosyl 5 phosphatase, which is this PIP5 enzyme here. We'll talk quite a bit about it today. So the big question that we have in the field is what determines the monogenic VSG expression in silence of all the remaining VSG genes at the same time? I have over 2,500 genes, only one transcribed at a time. What's the process regulating these mechanisms? And when you have a switching in VSG expression, so I have to take the active VSG gene from the expression site body, repress together with the other telomeres, and another telomere expression site in a different telomere has to be relocated within the nucleus for the site where transcription is occurring. So it's quite a big rearrangement. So this process must be regulated in some degree. And what's regulating this process? Now, what controls the switching in VSG expression? So we came across with the phosphoinositide signaling pathway a few years ago, and as a system regulating VSG gene expression, we have identified that these parasites have most of the enzymes involved in synthesis of those molecules, as well as those involved in the signaling process. When I talk about signaling, I'm talking about kinases and phosphatases, where the synthesis, the machinery required to produce those metabolites. And once you look at across evolution, you can see that archaea and bacteria have only the molecules involved in the synthesis of these metabolites. If you look now, trypanosomes, which are single-cell organisms, protozoa, Saccharomyces, plant, and humans, you can see they all have the signaling components, but more important, as you get into metazoan, which are more complex organisms, you have an expansion of those kinases and phosphatases enzymes. So we have just started to measure those metabolites in the cell, but we started our work with working on the enzymes. We made several mutant parasites lacking many of those enzymes, different individual knockout cell lines for each enzymatic step. And we found it here that many of those were essential for infection of mice. If you knock down, here's in a system in which you have a tetracycline regulated expression of the enzyme on the parasite, remove endogenous alleles of a particular enzyme. We add an allele that can be turned on and off in the presence of tetracycline, or its analog doxycycline. So we infect mice, we give water to the mice with or without doxycycline after we establish infection, and we look what happens to the infection once we remove doxycycline, meaning once you shut off the expression of that gene, shut down the expression of that gene. So in the white, similar to the white type situation, doxycycline plus, we'll get a normal infection. If you now remove doxycycline, we have a knockdown of the enzymes and parasites fail to infect the mice, which means that the system is functional and it's important for infection. There's below here's a few no mutants that we also tested. We just started to measure some of those metabolites. This is very preliminary data from our lab now. We are using targeted metabolomics to identify those metabolites. They are in very small quantities. We are growing here 10 to the 9, 10 to the 10 cells. So 10 is 4 plus liters of cells to be able to detect those metabolites. That's been a bit of the challenge to detect them. But our preliminary data here indicates that we have relatively higher amounts of the precursors, the phosphonosietide, phosphatidinosyl phosphates, and then PI45P2, PI345P3, then you have at least 100-fold less than the precursor. And we are also starting to measure the soluble inositol phosphates, but I won't show that to you today. But it's just to let you know that it's starting to get our hands on those metabolites and understand which lipids are associated to those molecules as well. But what's from the point of view of gene regulation, what was important and what got our attention to this path was the fact that as we knock down those genes, we got a transcription of all VSG genes at the same time, which is a contrast to the typical situation. Only one VSG is transcribed at the same time, at a single time. Now we have multiple of them, and this is by knockdown of either the PIP5K or the PIP5PA. So those are kinases that synthesize the molecule PIP3, PI45P2, PI345P3, and the phosphatase dephosphorylated these molecules. And then we see also a massive switching in VSG expression. That changed quite a bit our understanding of antigenic switching in trypanosomes. We always thought that this process was occurring in a stochastic fashion. Now the data started to raise the question that we may have a signaling system regulating this process. So we started to dig a little bit more into the process. Looking at the localization, we found here that PIP5K and PLC, as any other system, primarily at the plasma membrane of the organism, and the endosomes as well, or the organelles inside of the cell, but primarily on the membrane. But to our surprise, the phosphatase, which dephosphorylates PI345P3, we know that from enzymology and analysis with the purified enzyme, dephosphorylates the 5-phosphate here, PIP3 into PIP2. This enzyme localizes in the nucleus, and to our surprise, it was actually colocalizing almost perfectly with RAP1, which repressor activator protein 1. At the time, the only protein we knew was involved in the repression of VSG genes. Okay, so to study this system, and we felt that we should pull down the complex and the interactions and started to analyze what are the proteins involved in the process here. So, but what we notice is that if you knock down the PIP5BAs, the smallest migrator is a 0.1 megadalt in a native gel, which is an indicative of a formation of a protein complex. But if you knock down the phosphatase, the RAP1 associated protein disappears. So suggesting that the complex falls apart. So we decided to take a different approach. We generated 2-point mutations on the enzyme, which eliminate catalysis. So the enzyme cannot dephosphorylate PIP3 anymore and preserve the integrity of that complex. Complex still migrate as a 0.1 megadalton. That gave us the idea that interactions are most likely to be preserved, but the function is being eliminated. The enzyme cannot dephosphorylate anymore. Then we were able to study the function of the enzyme, and as a consequence, the role of this metabolite in antigenic variation. So to approach this we've been doing cross-linking mass spectrometry to map the interactions of the different molecules. So in this approach, we use DSS, which is an in vivo cross-linker, has a space arm of 11.4 angstrom with 2 reactive groups. If you have 2 amino acids within that proximity of 11 angstrom, which indicated they could be interacting, so the cross-link will react with primary amines, lysines, residues, for example, and cross-link the 2 peptides of the proteins, the 2 proteins. And when you do mass spec, typically what we identify is 2 amino acids with a cross-link. Here we have a lysate non-cross-linked and cross-linked. With the cross-link, we have a shift, which is visualized quite well here on the Western blot of the input and the L-weight. If you don't cross-link, you have this Western for the PIP5BAs protein. We get a single band. If we cross-link, we get a smear. Then you take this material, put down, put in the mass spec. We identify thousands and thousands of cross-linked peptides. When we look at the network, now we have been able to map several protein interactions now at peptide resolution. We know which protein is interacting to the other protein, and we know which peptide is involved in this interaction. That doesn't mean that we are capturing everything, because we could still have interactions that was not captured by virtue of the cross-link used. But it's giving us quite a good picture of the network that's happening there, mapping which proteins interacting with which protein here. Just to give an example here, I will look closely to this subnetwork. And if you look closely here, we have network containing RAP1 and PIP5BAs here. We always thought these 2 proteins are interacting directly based on all the mass spec data we did, quantitative mass spec, SciLike, all that stuff that it did before. But this data is actually suggesting that it doesn't say that it's not interacting directly, but you have another protein here actually functioning as a hub. Connecting to different proteins and keeping all this stuff together, which is NOP136. NOP136 is typically a nucleolus proteins, but is also a protein involved in the assembly of the nucleolus. We're getting a lot of proteins involved in the assembly of this type of structure, and you think that right now that these molecules might have a role in assembly that repressive telomeric cluster that we see in parasites when you do fish. So with this data, we can see where the interactions are occurring, any terminals to the proteins, the terminals interactions in the middle of the protein. We can map the peptides, which is kind of a quite useful type of information. This is just a closer look on the regions of the interactions that we get with this approach. As this parasite goes to the insect stage, they shut down all the VSG genes. So they change the surface coat to something that will adapt to the insect stage. So for this becomes very useful for us because all the VSG genes are shut off. They are not expressed. Then we decided to look, well, is this enzyme and this network of interactions involved in this repression in the bloodstream form, the infectious forms in animals and humans, or is also important for repression as the parasite developed to a non-infection form. So we pull down from the insect stage, we map the interactions. They have very similar composition on the interactions as we saw before. Get a lot of the nuclear pore proteins here, which we believe it is probably attaching to the nuclear lamina of the cell, of the parasite. But if we knock down now this enzyme, we get all the VSG genes transcribed even in the insect stage, which typically do not express VSG genes. So it's really a system important for repression of those genes. Let me just check the time. Yeah, I'm on time. Once we do fish and immunofluorescence analysis, you can see here for POL1, typically we get in a wild type cell is that POL1 stains the nucleolus and the expression site body, which is this tiny little dot here. This is where the VSG gene is transcribed. You can see on the combination of fish and IFA here. So only one VSG transcribed, POL1 is in a single site, which is as shown here in this diagram. Now, if knock down the phosphatase, we get multiple POL1 going to multiple expression sites. Now, what we think that is going on is this, and that's what is studying, revisiting a little bit of this data that we published before, is that this complex is functioning in the assembly of this compartment here to repress all the VSG genes. Is much less energy for the cell to dedicate a subset of proteins and group all these telomere expression sites together and repress them at the same time. But once we knock down, we get them all separated and expression occurs and switching occurs. So once we look at the RNA-seq to look at we have POL1 on those sites. We also have transcription. So we do have transcription on the silent expression sites. You can see here in the wild type with the VSG gene, there is no transcription. In the mutant, like in phosphatase activity, you get transcription now going on. On the active VSG genes gets down-regulated, and eventually you get a lot of cells that switch VSG gene expressed. Okay, in the wild type population, you get primarily VSG2 being expressed, and let me correct myself, in the population expressed in the wild type gene, okay, with a few other VSGs being transcribed. It's not a cell expressing multiple, but individual cells expressing different VSGs, and you get many of them expressing different VSGs here. To our surprise, we also get transcription of all the subtelomeric regions. If you look at the subtelomeric region, you get transcription through all those regions, which are typically silent in the cells, okay? We think there is a major organization of the chromatin here. We are doing POR-C, which is similar approach as high-C chromatin capture to conformation to analyze the organization of the chromatin, and we have some idea of what's going on here. Starting to get an idea of what's going on here. To evaluate the switching of the cells, we actually developed a new methodology, which is VSG-BAR-seq. So we can actually look at individual cells in a population. We barcode the cells and do nanopore sequencing. It's important for us to do nanopore sequence because VSG genes are very similar to each other, but with the nanopore, you get long reads that we can distinguish the different VSGs. So if you now express the mutant phosphatase, you get most of the cells expressed in a different VSG rather than the original one. And once we look at the sites where they are expressed, you can see that in the clone that does not switch, they express the VSG here. The clone that switch, you can see the reads are coming from a different VSG, from a different expression site. This is just to illustrate to you. to get a little bit of an insight of what could be going on. I mean, how this metabolite could be regulating so much of this process. So we went to look at the PIP5BA's partner, which is RAP1, a repressor activated protein. We know RAP1 was repressing. If you knock down, we get transcription. We had no idea of how this was happening, okay? So we decided to do ChIP-seq with RAP1. We found the RAP1 was binding to several places in the genome to AT-rich regions. It was not binding to the expression site, to the active expression site, um, which reinforces its role in repression of these genes, but it was binding to, especially to 7 base pair repeats and telomeric repeats, which are regions surrounding the VSG gene. So it's binding just around the VSG gene, not in the gene itself, and insulating the gene repressing those regions. As for the subtelomeric regions, we don't get much binding there, but we found that it binds to centromeres. Centromeres are also AT-rich regions in the cells, and which most of them are also, many of them are subtelomeric in the cells. So we went to validate those binding assays on the ChIP-seq. We found with recombinant RAP1, we make several recombinant protein here, RAP1 full length, and different domains of RAP1. The N-terminal domain, the MYB and MYBL domain. MYB is the DNA binding domain. MYBL is the MYB-like domain. Domains that we predicted would be binding to DNA. But in the process, we identified this VHP domain, the N-terminus of RAP1, which is a Villain headpiece domain. It turned out to be very important. So once we do binding assays, we saw RAP1 confirmed the interactions that were observed on the chip, that it binds to telomeric repeats, binds 7 base pair repeats. We could measure the kinetics of binding with macroscale thermophoresis. It's on the nanomolar binding affinities, which is a very, very strong interaction. If you look at each domain separately, we can see MYB binds very nicely to both 7 base pair repeats or telomeric repeats, as well as the MYBL domain. But the VHP does not bind to the DNA. But here's the interesting part of the story, okay? This VHP, we found it to be binding to the PI345P3 metabolite. So it was actually binding to this metabolite. We did these assays using biotinylated PIP3. And we suspect this binding is due to the VHP domain, which is, has been shown to bind PIP2. But in our case, it does not bind PIP2, binds to PIP3, does not bind to any other metabolites but PIP3, okay? So let me see my time. Yeah, a few minutes more, I'll be done. So we did some competition assays to determine whether these metabolites could be interfering with the RAP1 bind to the DNA. We can see that once widened this metabolite, it inhibited the DNA binding of this molecule. And this is the same one. We can measure the kinetics, and we see a massive shift of the RAP1 binding to the DNA in the presence of PIP3, but not PIP3, PIP2 for both telomeric repeats and 7 base pair repeats. So these metabolites functioning as a regulator of RAP1 binding to the DNA. To analyze that in vivo, we made ChIP-seq in a cell that's mutant for the phosphatase, cannot dephosphorylate the metabolite. So we predict an accumulation of this metabolite that will bind to RAP1 and remove RAP1 from the DNA, and that's what we see. We can see here a combination of ChIP-seq and RNA-seq. Once RAP1 is bound to the DNA, there is no transcription. VSG genes are silent. When RAP1 now, when the mutant is expressed, PIP3 accumulates. You can see that accumulation, especially on this plot here. When we measure RAP1 bound PIP3 in the white type, almost no PIP3 bound to RAP1, but once you express a mutant, you get the metabolite. In this experiment, we put down the protein, and then we measure the amount of metabolites associated to the protein, and you get a significant increase, 100-fold increase on this metabolite bound to the protein, okay? And that we get protein now being removed from the DNA and transcription going on. So what we believe now is this metabolite function as allostatic regulator in the nucleus, controlling the binding of the protein to DNA and repression and switching of VSG genes. So just to finalize for you, what I'm trying to do here is just to give an example of how those metabolites can play a significant role in the regulation of immune evasion sporocytes in the nucleus of the cell, functioning the regulation of VSG gene. So we identify a number of proteins, which call telomere expression site complex. We believe now that we're starting to investigate these. That's our hypothesis, to be honest. Not a strong belief yet, but that's what we are investigating. That these proteins function assembling a telomeric repressive compartment. This compartment here, as you knock down the P5 base, these all go apart. That silence VSG genes, and this compartment going apart associates with transcription. The PIP3 is an allostatic regulator of ROP1 and control telomeric ES transcription, repressing the VSG genes. And why is that important? Important because the system can function as an on-off genetic switch to control activation and silencing of these VSG genes, which is important for the periodic switching VSG gene expression during antigenic variation, as well as the development of the cell to the insect stage. As they go to the insect stage, they have to repress everything to keep VSGs silent, to switch the surface code. So where we are going now, we are looking at mechanisms underlying compartmentalization of the genome, trying to get a hand on the molecules, but also the DNA, how the DNA is coming together on those telomeric expression compartment, repression compartment. We are doing quite a lot of POSI analysis. I couldn't show this data here because of time. Oksana Kotova, postdoc in the lab, is working on these. We identify protein composition of the expression site body, which is involved in monogenic expression, and how the signaling is connected from the inside of the nucleus to the outside of the nucleus, right, in the cytosol or beyond there. That's what we're investigating right now. This is the team that's been working on these. We are the Institute of Parasitology at McGill University. It's a very beautiful site here at the McDonough campus.